There is a moment, deep in opioid withdrawal, that the body treats as a kind of dying. The mu-opioid receptors that fentanyl or heroin had flooded for weeks are suddenly empty, and every system that adapted to their presence rebounds at once. The sweating, the gooseflesh, the bone-deep ache, the racing heart, the certainty of a wretched week still ahead. This is dependence made physical. Neurons that down-regulated their own signaling to survive the drug, now screaming in its absence. And then, in the accounts that have followed this substance for 60 years, something strange happens. The person swallows a single dose of a bitter extract drawn from the root bark of a West African shrub, Tabernanthe iboga, and the molecule inside it, called Ibogaine. And within roughly a day, the withdrawal is simply gone. The craving too, not eased, gone. This is not folklore whispered around a fire. It is documented again and again across decades. But here is the mystery that will hold us tonight. The real question is not whether Ibogaine interrupts addiction. It plainly does something. The question is how. How a single molecule with no clean target, no tidy lock and key, a molecule that touches opioid and NMDA and serotonin and nicotinic receptors all at once, accomplishes what no precisely engineered pharmaceutical ever has. One dose, one day, and no one can fully say how. Because the same molecule that seems to reset the mind can also stop the heart. So let your shoulders drop, and let the day loosen its grip on your jaw and your hands. This is Pharmasomnia, where we take the machinery of the body and slow it right down, night after night, until the science itself becomes something you can fall asleep to. If these slow journeys through your own chemistry bring you any rest at all, you might let the channel know, with a like or a subscribe. And then we won't ask again, and we won't raise our voice for the rest of the long night. From here on, we only wander, and we only explain, softly, the machinery underneath your skin. Far inland from the Atlantic coast of Gabon, where the rainforest closes overhead like a cathedral of leaves, and the air is warm and thick and green, there grows a small shrub with pale flowers and orange fruit. Its name is Tabernanthe iboga. To a botanist, it is a member of the Apocynaceae, the same family that gives us oleander and the periwinkle. To the people of the Bwiti religion, who have gathered here for generations, it is something else entirely. It is the teacher. It is the door. It is the plant that lets the living speak with the dead. And beneath the poetry, there is chemistry, precise, molecular, and old. Tonight, we begin there, at the root. Picture the digging. The initiate, or those who tend the initiate, unearth the root of the iboga plant, and it is the bark of that root, scraped and dried and ground into a bitter grey-brown powder that holds the medicine. Not the leaves, not the flowers, not the fruit, the root bark. And that bitterness on the tongue is not incidental. Bitterness, in the botanical world, is very often the taste of alkaloids, nitrogen-bearing molecules that plants build as chemical defenses, and that, again and again across human history, turn out to bend the workings of the animal nervous system. Morphine is an alkaloid. Caffeine is an alkaloid. Nicotine, quinine, strychnine, all alkaloids. And the iboga root holds not one, but a family of them. The chief among them, the one that will occupy the whole of our long night, is called Ibogaine. Its formal chemical name is 12-methoxyibogamine, and if you could hold its structure in your mind's eye, you would see, at the center of it, a familiar ring, a double ring, in fact, a 6-membered ring fused to a 5-membered ring, with a single nitrogen atom tucked into the smaller one. This is the indole. Remember that word, because it is the thread that runs through the whole story. The indole is the same molecular skeleton that sits at the heart of serotonin, the neurotransmitter of mood and satiety and sleep. It is the same skeleton inside tryptamine, inside psilocybin from the mushroom, inside DMT, inside the LSD molecule. Ibogaine is not itself a simple tryptamine, but it is built on that same indole foundation, dressed in extra rings and a methoxy group hung off one edge. It is, in a sense, a distant structural cousin of the serotonin your own brain is making right now, as you lie here and slow your breathing. And that structural resemblance is not a coincidence of no consequence. In pharmacology, shape is destiny. A receptor is a protein folded into a pocket, and a molecule fits that pocket, or it does not, the way a key fits a lock, and the closeness of the fit decides whether the door opens. Because ibogaine carries the indole shape that serotonin carries, ibogaine can drift into places in the brain that were built for serotonin, the serotonin receptors, the serotonin transporter, and press against them and be mistaken, partly, for the real thing. Hold that thought gently. It is the first clue in a mystery that, even now, remains unsolved. We are foreshadowing the transporter called SERT, the little molecular pump that recycles serotonin out of the synapse, and we will return to it again and again as the night deepens. But first, the ritual, because the chemistry was discovered inside the ceremony, not the other way around. In the Bwiti tradition, Iboga is taken at the great threshold of a life, the initiation. A person who wishes to become a member, to cross from the ordinary world into the world of the ancestors, consumes Iboga not in the small, careful amounts that dot Western use, but in massive quantity, spoonful after spoonful of the ground root bark, eaten over hours, in doses that would be reckless anywhere else on earth. The bitterness is overwhelming. The body rebels. Nausea comes early and stays. And then, slowly, the alkaloids cross from the gut into the blood, and from the blood into the brain, and the initiate is led down into an ordeal that can last a full day and a full night and beyond. It is not, by the accounts of those who have lived it, a pleasant euphoria. It is work. It is a long, immersive, waking dream, a flood of vivid imagery, of memories surfacing unbidden, of the sensation of reviewing one's own life as if from outside it, of encounters described as meetings with the dead, with ancestors, with the teacher itself. The Bwiti do not describe a high, they describe an audience. The plant, they say, tells you things. And through the long hours the initiate lies still, guided and watched over, while the visions run their course and the body labours under the chemical load, until at last, near the far edge of a day, the tide begins to recede. This is the image I want to leave with you, the anchor we will come back to through every chapter that follows. Do not think of Ibogaine as a hand pressing a single button in the brain. Think of it as an uninvited teacher who walks into a great house and does not stop at one room. It moves down the corridors. It speaks in the doorway of one chamber, and then another, addressing many parts of the mind at once. The serotonin rooms, the dopamine rooms, rooms we have not named yet, rooms with locks we still cannot pick. Most drugs whisper to one receptor. Ibogaine holds a conversation with the whole house. And that, more than anything, is why it can do what almost nothing else can do, and why almost nothing else is so hard to understand. For now, keep the teacher walking those corridors, and let us follow the molecule the way the body follows it, through the mouth, into the gut, into the blood. Here we meet the first hard, humbling fact of Ibogaine's pharmacology, and it is a fact of the plumbing rather than the mysticism. Ibogaine is absorbed poorly, and it is absorbed unpredictably. Swallow the root bark, and only a portion of the alkaloid crosses the wall of the small intestine into the bloodstream, and the portion that crosses varies from person to person and even from occasion to occasion. The word pharmacologists use for the fraction of a swallowed dose that actually reaches the circulation intact is bioavailability, and Ibogaine's oral bioavailability is low and erratic. This is part of why the ceremonial doses are so large. Much of what is eaten never truly arrives. And of the fraction that does slip through the intestinal wall, much is intercepted almost immediately. The blood leaving the gut does not run straight to the brain. It runs first through the liver, the great chemical customs house of the body, and the liver is armed with enzymes whose entire purpose is to seize foreign molecules and take them apart. This interception at the very first pass through the liver is called first-pass metabolism, and Ibogaine is subject to it heavily. A swallowed molecule of Ibogaine must survive the gut, then survive the liver, before it ever reaches the neurons it will teach. Many do not survive. They are transformed on arrival. And here is where the story turns strange in a way that will matter enormously later, so let it settle in slowly, the way the alkaloid itself settles into tissue. The liver enzyme that does most of the transforming belongs to a vast family called cytochrome P450, the body's principal machinery for chemically dismantling drugs, a whole workshop of related enzymes, each tuned to particular molecular shapes. The specific member that acts on Ibogaine is called CYP2D6. Say it slowly. CYP2D6. It is one enzyme among dozens, but it is the one whose hands are on this molecule. What CYP2D6 does to Ibogaine is a small chemical edit. Remember the methoxy group, that little oxygen and carbon appendage hung off the indole framework, the very thing named in the word methoxyibogamine. CYP2D6 removes the methyl from it. It performs what chemists call an O-demethylation, snipping off a single carbon-hydrogen cluster and leaving behind a bare hydroxyl, an oxygen bound to hydrogen, in its place. It is a tiny alteration, one small group swapped for a smaller one on an enormous molecule. And yet, by that single edit, Ibogaine becomes a new compound entirely. That new compound is called noribogaine. I will only introduce noribogaine tonight. We will not yet explore all it does, because it deserves its own long stretch of the night. But understand what has just happened, because it overturns the ordinary way we tell drug stories. Usually, the body breaks a drug down in order to switch it off. Metabolism as disposal, the liver reducing an active molecule to inert fragments the kidneys can flush away. Here, that is not what happens. When CYP2D6 converts Ibogaine into noribogaine, it does not extinguish the drug. It creates a second active drug, a metabolite that lingers in the body far longer than its parent, that binds serotonin machinery even more avidly, that may carry a great deal of the long, quiet after effect. The liver, meaning to disarm the teacher, has instead sent a second teacher walking down the corridors. And so, a single swallow of bitter root gives rise to two overlapping presences in the brain. Ibogaine first, fast and strange, and noribogaine behind it, slower and more patient, outlasting the visions by days. There is one more thread here worth holding, because it explains why no two initiates meet quite the same teacher. The gene that builds CYP2D6 varies enormously between people. Some of us carry versions that build fast, hungry enzyme, extensive metabolizers they are called, who convert ibogaine to noribogaine quickly. Others build sluggish or scarce enzyme, the poor metabolizers, in whom ibogaine lingers longer as itself before it is transformed. This is not a small footnote. It means the very ratio of the two teachers in the brain, the balance of the fast, strange presence against the slow, patient one, is written differently in each person's DNA. The same spoonful of the same root, given to two people side by side, becomes two different chemical experiences because their livers speak different dialects of the same enzyme. It is a warning we will honor all night. This is a molecule of variation, of unpredictability, a drug that resists the tidy dose-response curves that pharmacology loves. So let us gather what we hold so far and set it down where we can find it again in the dark. There is a plant in the forests of Gabon, Tabernanthe iboga, whose root bark is bitter with alkaloids. Chief among them is ibogaine, formerly 12-methoxy-ibogamine, built upon the indole scaffold, the same molecular skeleton at the heart of serotonin. Our first quiet hint that this molecule will meddle with the serotonin system and its recycling pump, SERT. Swallowed in the great initiation rite of the Bwiti, in doses that would astonish a Western clinic, ibogaine is absorbed poorly and unpredictably, is stripped hard on its first pass through the liver, and is there converted by the cytochrome enzyme CYP2D6, through the removal of a single methyl group, into a second, longer-lasting active molecule, noribogaine. Two teachers, then, walking the corridors of the house, their proportions decided by a gene that no two of us carry quite the same way. And here is the spine of everything to come, the sentence I will return to more times than you can count before the night is over. This ancient rite, this bitter root, eaten in a forest clearing under a religion far older than pharmacology, this is the very same molecule that modern medicine has watched, with astonishment, interrupt the deepest addictions we know, and that modern medicine still cannot fully explain. The Bwiti met the teacher long before we knew its name. We have named it, mapped its rings, identified the enzyme that transforms it, measured its poor absorption and its long half-life. And still, when we ask the teacher its most important secret, how it does what it does to the addicted brain, the answer does not come cleanly. That is the mystery we will walk toward, slowly, over the hours ahead. Tonight, we have only stood at the root, at the bitter bark, and the day-long vision, and the small chemical edit that makes one molecule into two. It is enough to begin with. Let the image of the teacher walking the corridors stay with you as your breathing lengthens, a molecule that does not knock on one door, but speaks softly in every room at once, in the serotonin rooms, and the dopamine rooms, and the opioid rooms, and the corridors between. Let that image carry you forward, into the year 1962, into a small apartment in New York, into the story of a young man who did not know he was about to become the accidental father of a field. His name was Howard Lotsof. He was 19 years old, and he was addicted to heroin. This is where the story of Ibogaine as medicine truly begins. Not in a laboratory, not in a clinical trial, but in the ordinary desperation of a young man in a city, looking, as so many of his friends were looking, for the next interesting thing to put into his body. A chemist friend had given him a white powder, Ibogaine, isolated from the root of that African shrub we spoke of, the Tabernanthe iboga. Lotsof took it, expecting a psychedelic, expecting colors, expecting hours of strange interior cinema, expecting, perhaps, something like the mescaline or the LSD that moved through his circle in those years. He got the long trip, 30 hours or more of it, the dreamlike waking vision that Ibogaine famously produces. And then, when it finally released him, he noticed something that made no sense at all. He was not sick. To understand why those three words are extraordinary, why they are, in a sense, the entire reason we are here tonight, you have to understand what should have happened to Howard Lotsof's body in the hours after his last dose of heroin. You have to understand withdrawal, not as a vague misery, but as a precise molecular event, a storm with named streets and named weather. So let us walk into that storm slowly, while you lie still and safe and warm, and learn the shape of the thing that Ibogaine impossibly switched off. Heroin, once it crosses into the brain, is rapidly converted to morphine, and morphine goes looking for a particular door. That door is the mu-opioid receptor, a protein embedded in the membranes of neurons throughout the brain and spinal cord and gut, coiling 7 times through the cell wall, coupled on its inner face to a G protein. When morphine binds and turns the key, that G protein dissociates and fans out to do several quiet things at once. It closes calcium channels, so the neuron releases less of its signaling chemicals. It opens potassium channels, so the neuron grows harder to excite, harder to fire. And crucially, it reaches inward to an enzyme called adenylyl cyclase and switches it down, so that the cell makes less of a small, vital messenger molecule called cyclic AMP, CAMP. Less calcium in, more potassium out, less CAMP made. The neuron goes quiet, pain fades, breathing slows, and in the reward pathways, dopamine surges, and the body is flooded with a warmth it did not earn. That is the high. That is the door opened. But the body is not a passive thing. The body is a homeostat, always leaning against whatever pushes it, always seeking the level. And when the mu-opioid receptor is turned that far down, that often, for that long, the neuron begins, patiently, to fight back. This is the true birth of addiction, and it happens in slow, molecular increments. First, the receptors themselves begin to change. With relentless agonism, relentless binding of morphine, day after day, the mu-opioid receptors become desensitized. They uncouple from their G proteins. They are pulled inward, off the cell surface, in a process called receptor internalization, down-regulated, fewer of them left facing the outside world to answer the knock. This is tolerance in its molecular form. The same dose speaks to fewer receptors, and so the same dose says less, and the person needs more heroin simply to reach the place the first dose reached so easily. More knocking at fewer doors. And beneath the receptors, in the machinery the receptors command, something more dangerous is happening. Remember that the opioid was pressing adenylyl cyclase down, suppressing CAMP. So the cell, leaning against that suppression, does the opposite. It builds more adenylyl cyclase, ramps up the whole cyclic AMP pathway, upregulates it, primes it like a spring held down under a heavy hand. As long as the opioid is present, this hidden overcompensation is invisible. The upward push of the cell and the downward push of the drug cancel almost perfectly, and the person feels, more or less, normal. They are not normal. They are a balance held between two enormous opposing forces, and they no longer know it. They have become, in the truest sense, dependent. The drug is now required simply to hold the equilibrium the drug itself created. And then the drug leaves. Imagine the hand lifted suddenly off the held spring. This is withdrawal, and now you can see it for what it is at the level of the molecule. The heroin clears the mu-opioid receptors, but the compensations do not clear. The upregulated adenylyl cyclase is still there, still overbuilt, and with nothing left to suppress it, cyclic AMP floods upward, everywhere, all at once. Neurons that had been held quiet for months come roaring back overexcited, firing far beyond their baseline. The homeostat that leaned so hard against the opioid is now leaning against nothing, and it topples. Every system that the opioid had slowed now overshoots into the opposite. The gut, whose motility opioids famously freeze, cramps and races. The diarrhea, the nausea. The pupils dilate. The skin crawls and pricks with gooseflesh, the old word for it, cold turkey. The body's thermostat lurching. The muscles ache and twitch and kick, and at the center of the physical agony sits one small structure with a beautiful name, the locus coeruleus, the blue place, a tiny knot of neurons in the brainstem, the single largest source of noradrenaline in the entire brain. In the quiet times, opioids restrain the locus coeruleus, hold its firing down through those same G-protein hands, that same cyclic AMP suppression. So the locus coeruleus, too, has been leaning back against the drug, its own CAMP pathway upregulated, its own spring wound tight. When the opioid is withdrawn, the locus coeruleus fires in a storm. Noradrenaline pours out across the brain in a flood, and this noradrenergic storm is the engine of the worst of it. The racing heart, the sweating, the surging blood pressure, the anxiety that rises to terror, the sleeplessness, the profound cellular sense of wrongness that makes a person do almost anything to make it stop. This is why the treatment for the physical storm has often been a drug like clonidine, which calms exactly that structure, an alpha-2 adrenergic agonist that presses down on the locus coeruleus and dampens the flood. That is withdrawal, not weakness, not merely a bad feeling, a precise, predictable, molecular rebound. Receptors down-regulated, cyclic AMP overbuilt, the blue place unrestrained and roaring. It arrives on a schedule you can almost set a clock by. For heroin, the first shadows of it appear within 6 to 12 hours of the last dose, deepen through the first day, and peak somewhere around the second or third day. It is one of the most reliable events in all of pharmacology. Take an opioid-dependent body, remove the opioid, and the storm comes. It always comes. Except it didn't come for Howard Lotsof. He took his Ibogaine. He rode the long, strange vision for 30 hours. And when he came down and looked for the storm, the 6-hour shadows, the cramping gut, the roaring blue place, it simply was not there. His withdrawal had not arrived. And more than that, more astonishingly than that, his craving was gone. Not muted, gone. He looked at the heroin, the substance that had organized his entire life around itself, and he felt, by his own account, no desire for it at all. He described later the sudden realization that he had been given a tool to end his addiction to narcotics, that the thing that had held him was simply, quietly, no longer holding. Now, sit with how strange that is, because the strangeness is the whole point, and it is the thread we will follow for the rest of these hours. Ibogaine is not an opioid. This is not a small technicality, it is the heart of the mystery. When we treat opioid withdrawal with methadone or with buprenorphine, we are doing something conceptually simple, something the molecules explain cleanly. Methadone is itself a mu-opioid agonist. It walks up to the very same door heroin used and opens it, more slowly, more steadily, with a long half-life that smooths the peaks and troughs, but it is fundamentally a substitution. It keeps the held spring held. It keeps the compensated cell compensated. Buprenorphine is a partial agonist at the same receptor, plus an antagonist at another opioid receptor, the kappa. It, too, occupies the mu door, but only opens it part way, enough to quiet the storm without the full flood of reward. Both drugs work by speaking the opioid's own language, at the opioid's own receptor. Take methadone away, and withdrawal will eventually come from the methadone. The debt has been transferred, not cancelled. Ibogaine does none of this. Ibogaine has, at best, only weak and complicated interactions at the mu-opioid receptor. It does not sit down in that chair and impersonate morphine. It does not keep the spring held. And yet the spring, somehow, did not fire. The storm that should have come, on its reliable clock, from that overbuilt cyclic AMP machinery and that unrestrained blue place, the storm did not come. Something in that long day of vision reached into the addicted brain, and without occupying the mu-opioid receptor as a substitute, without transferring the debt to a new drug, appeared to reset the very compensations that make withdrawal inevitable. The upregulation quieted, the rebound prevented, the craving erased. And the tool that did it was gone from the body within a day or two, leaving no need for a daily dose, no maintenance, no substitute chair to sit in. One dose, one day. And no one can fully say how. Let that refrain settle into you, because we will return to it again and again as the hours pass. One dose, one day, and no one can fully say how. It is the sentence that has haunted this molecule for 60 years, ever since a 19-year-old in a New York apartment noticed the absence of a storm that should have arrived, and did not. And here, before you drift further, let us set down the inventory, the running list of things we will need to explain, the questions we are carrying with us into the dark. Because Lotsof's accident did not give us an answer. It gave us a set of impossibilities to account for. Withdrawal, gone. The physical storm of the upregulated cyclic AMP pathway and the roaring locus coeruleus, somehow prevented, without an opioid to hold it down. That is the first item. Craving, gone. The deep, learned hunger of the reward system, the dopamine-driven wanting that outlasts the physical sickness by months or years and drives most relapse, somehow erased or reset in a single day. That is the second item. And all of it accomplished without opioid replacement, without a substitute agonist at the mu receptor, without methadone's held spring or buprenorphine's partial key, so that whatever Ibogaine did, it did by some other road entirely. That is the third and the strangest. 3 impossibilities: withdrawal gone, craving gone, and no opioid in the chair. Hold those 3 the way you might hold 3 small stones in your palm as you fall asleep. We will take each one out and turn it over in the light of real receptors and real enzymes in the hours to come. We will ask what Ibogaine touches when it is not touching the mu opioid receptor as a substitute. We will follow it into the serotonin rooms and the dopamine rooms, and to a strange receptor called sigma, and to the NMDA channel, and to the quiet machinery inside the cell that might, just might, explain how an overbuilt cyclic AMP pathway could be talked down in a single night. And we will meet the metabolite, the second molecule that the body carves out of the first, that lingers long after Ibogaine itself is gone, and may be the true agent of the long quiet that follows. But all of that is ahead of us. For now, tonight, it is enough to stand where Howard Lotsof stood, in that apartment, in that year, in that astonished silence where the storm should have been and simply was not. He was not a scientist. He had no framework for what had happened to him. He only knew what his body was telling him, in a language older than any pharmacology. The wanting was gone. And that absence, that strange, unearned stillness, would become the question that has occupied laboratories for the better part of 40 years since. So let us begin to open the molecule itself, slowly, the way you might unfold a letter you have read many times but never quite understood. Because here is the difficulty that greets everyone who tries. Most drugs, when we teach them, can be explained with a single sentence and a single lock. Morphine and heroin and oxycodone fold themselves into the mu opioid receptor, and that one act, that snug fit of molecule into binding pocket, accounts for the warmth, the analgesia, the slowed breath, the dependence. A benzodiazepine such as diazepam slips into a quiet corner of the GABA-A receptor and, without opening the channel itself, makes that channel more responsive to the brain's own calming signal. And from that single allosteric touch flows the drowsiness and the ease. Cocaine wedges itself into the dopamine transporter and blocks it, so that dopamine cannot be swept back out of the synapse, and the whole euphoric flood follows from that one obstruction. One drug, one target, one story. That is how pharmacology prefers to speak. That is the sentence the textbook wants to write. Ibogaine refuses the sentence. It refuses it completely, and that refusal is not a footnote to the mystery. It is the mystery. Because Ibogaine does not carry a single key. It arrives at the nervous system with a fistful of them, and it presses on many locks at once, and no one has ever been able to say with certainty which of those pressures, or which combination of them, is the one that quiets the addicted brain. Think of it not as a key sliding cleanly into one lock, but as a hand laid flat across a wall of switches, pressing a dozen of them down together. Some hard, some barely, some in ways we have measured precisely, and some in ways we can only estimate. Every channel, every transporter, every receptor it touches all at once, and the effect that emerges is not the sound of one switch, but the chord of all of them sounding together. A drug with no single mechanism cannot be explained by any single mechanism. Hold that sentence close tonight. We will return to it again and again, because it is the whole shape of the problem. So, let us walk the wall of switches, one at a time, and name each one, and press it down slowly, and listen to what it does. The first switch, and the one we understand best, is the NMDA receptor. This is a glutamate receptor, glutamate being the brain's principal excitatory messenger, the molecule that says, fire, connect, remember, across the vast majority of the brain's signaling synapses. The NMDA receptor is a particular and beautiful kind of gate. It is a channel that lets calcium and other ions flow into a neuron, but it is doubly locked. It will not open unless glutamate binds it, and the cell is already somewhat depolarized, already leaning toward activity, so that a magnesium ion normally plugging the pore is expelled. Because it demands both signals at once, the NMDA receptor is the brain's great coincidence detector, the molecular device that notices when two things happen together and strengthens the connection between them. It is, in the most literal cellular sense we possess, a machine for association. It is how the smell becomes the memory, how the place becomes the fear, how the ritual becomes the craving, and Ibogaine blocks it. It blocks it in a specific and telling way, as what pharmacologists call an uncompetitive open-channel blocker. This means Ibogaine does not fight glutamate for its binding site. It does not jostle at the door. Instead, it waits for the channel to open in the ordinary course of business, and then it slips down inside the open pore and lodges there, physically obstructing the flow of ions through a gate that is already ajar. It is the same broad mechanism by which ketamine works, and by which the old dissociative anesthetic phencyclidine works. And this shared mechanism is why Ibogaine, at the doses that interrupt addiction, carries with it that dreamlike, dissociative, waking vision quality, the oneirogenic flood we spoke of earlier. Uncompetitive because it needs the channel open to act. Open channel because it acts from within the pore itself. Say those words slowly to yourself, and you can almost feel the shape of it. A molecule drifting into an open gate and quietly sitting down in the doorway. Now here is why that matters for the storm that would not come. If the NMDA receptor is the brain's machine for association, for welding one thing to another, the cue to the reward, the needle to the relief, the corner of the street to the pull in the chest, then a drug that dampens the NMDA receptor is a drug that loosens the welds. Glutamatergic transmission through NMDA channels is deeply implicated in the way addictive learning is laid down and, crucially, in the way it is maintained and reconsolidated each time it is recalled. Block those channels, quiet that glutamate signaling for a night, and you may catch the addicted brain in the act of rehearsing its own compulsion and interrupt the rehearsal. This is the most concrete, best characterized guess we have for how a single dose might reach in and cut a wire that took years to lay. It is not the whole answer, but it is the switch we can see most clearly, and it is pressed hard. Let us move our hand to the next switch. This one is stranger, and its story runs in two directions at once, toward healing and toward harm. It is the sigma-2 receptor. The sigma receptors were, for a long time, one of pharmacology's small embarrassments. Proteins that bound certain drugs avidly and yet resisted easy classification. Orphans that did not fit the familiar families. The sigma-2 receptor has now been identified with a protein called TMEM97, sitting in the membranes of the cell, entangled with cholesterol handling and with the machinery of cellular stress. Ibogaine binds it with real affinity, and here the story forks. On one hand, sigma-2 activation is strongly implicated in ibogaine's darkest property, its neurotoxicity. In animal studies, high doses of ibogaine kill Purkinje cells, the great branching neurons of the cerebellum, in a pattern that tracks with sigma-2 engagement. This is not a small thing to hold in the dark. The same molecule that may loosen the welds of addiction may, at the wrong dose, damage the very neurons it moves through. But the sigma receptors are also woven into neuroplasticity, into the cell's capacity to grow, to sprout new connections, to remodel itself. And so, the same switch that carries the warning may also carry part of the promise. The possibility that ibogaine does not merely interrupt a signal for one night, but opens, briefly, a window in which the brain becomes unusually willing to rewire. Two directions, one receptor. Healing and harm pressed down by the very same fingertip. We do not yet know how to have the one without the other, and that uncertainty is not a flaw in the telling. It is the honest edge of what is known. Move again. The next switch belongs to a molecule you have met before in gentler company, serotonin. Ibogaine's core structure is an indole alkaloid, and the indole ring is the same molecular scaffold that sits at the heart of serotonin itself, and of the tryptamine psychedelics, and of the triptans, and of a great scattered family of mind-shaping compounds. That kinship is not decorative. It has consequences. Ibogaine inhibits the serotonin transporter, SERT, the pump embedded in the membranes of serotonergic neurons whose ordinary job is to reach back into the synapse after a signal has fired and draw the released serotonin back inside, ending the message and recycling the molecule. This is precisely the pump that the SSRI antidepressants, fluoxetine, sertraline, the whole familiar shelf of them, block for weeks and months at a time to lift depression. Ibogaine blocks it too. When that reuptake pump is obstructed, serotonin lingers longer in the synaptic cleft, its signal prolonged and amplified, the serotonergic tone of the brain raised. Some of ibogaine's mood-altering, perception-altering character almost certainly flows from this raised serotonergic tide, this echo of its indole ancestry pressing on the same machinery the antidepressants press on. Though ibogaine does it acutely, all at once, in a single overwhelming night rather than a slow chemical season. And there is a caution folded inside that switch, one worth naming even here in the quiet. A drug that raises synaptic serotonin, taken alongside other drugs that raise synaptic serotonin, the SSRIs, certain opioids like tramadol, the older MAO inhibitors, can push serotonergic signaling into a dangerous excess, the state clinicians call serotonin syndrome. It is one more reason that ibogaine is not a molecule to be met carelessly. But tonight, we are only naming switches, and this one is real. SERT, inhibited, serotonin pooling, the indole scaffold quietly betraying its family resemblance. One more switch, and then we will rest our hand and consider what the chord sounds like altogether. This last one may be the most quietly important for understanding Ibogaine's peculiar breadth. It is the nicotinic acetylcholine receptor, and in particular, the subtype built from alpha-3 and beta-4 subunits, written in the shorthand of the field as the alpha-3 beta-4 nicotinic receptor. Acetylcholine is another of the brain's messengers, and these nicotinic receptors are the very channels that nicotine itself seizes upon, the receptors that make a cigarette a cigarette. Ibogaine, and even more potently its metabolite, act as antagonists at the alpha-3 beta-4 subtype. They occupy or block these receptors, dampening their signaling. And here is the remarkable part. The alpha-3 beta-4 receptor is richly present in a small, ancient midbrain structure called the medial habenula and its downstream partner, the interpeduncular nucleus, a circuit increasingly understood as a kind of brake, a region that generates the aversive, unpleasant dimensions of withdrawal and that helps govern the drive to seek across many different drugs. That last phrase is the key, across many different drugs. Because most anti-addictive medicines are narrow, methadone and buprenorphine speak only to the opioid system. Varenicline was built for nicotine alone. But Ibogaine's reputation, however hard it is to verify, is for breadth. Reports of it quieting the pull of opioids and stimulants and alcohol and nicotine alike. A single receptor tuned to one drug cannot explain a breadth like that. But a receptor sitting in a shared circuit of craving and withdrawal, a switch wired into the common trunk rather than one of the branches, just might. Antagonism at the alpha-3 beta-4 nicotinic receptor is our best current candidate for why Ibogaine seems not to care which drug it is you cannot stop taking. It presses on the trunk, not the leaf. So now, rest your hand flat against the wall and feel how many switches are pressed down at once. The NMDA receptor, blocked from within its open pore, its association machine dampened. The sigma-2 receptor bound, carrying both the promise of plasticity and the threat of harm. The serotonin transporter inhibited, serotonin pooling in the synapse, the indole ancestry showing through. The alpha-3 beta-4 nicotinic receptor antagonized, the shared circuit of craving quieted at its trunk. Every channel, every transporter, every receptor it touches all at once. And these are only the ones we have measured with confidence. There are more, fainter pressures we have not named tonight. Touches at the kappa and mu opioid receptors, at other sigma sites, at other transporters, a whole penumbra of weaker affinities surrounding the strong ones like a blur around a struck bell. And this is the difficulty and the beauty and the whole shape of the problem. When morphine acts, we can point to the mu opioid receptor and say, there, that is the cause, that single snug fit. When cocaine acts, we point to the dopamine transporter and say, there. But when Ibogaine acts, when the storm does not come, when the wanting simply lifts and stays lifted for weeks, we cannot point to any single place and say, there. There is a second character in this story, and it arrives quietly, after the lightning has already struck. Because Ibogaine, for all its dramatic entrance, does not stay long. Once it is swallowed and absorbed, once it crosses out of the gut and into the blood, it meets an enzyme waiting in the liver, a member of the cytochrome P450 family called CYP2D6. And CYP2D6 does to Ibogaine what it does to so many molecules that pass through us. It modifies it. Specifically, it performs a reaction called O-demethylation. Ibogaine carries a small chemical decoration, a methoxy group, an oxygen bonded to a methyl cap, that little cluster of 1 carbon and 3 hydrogens. CYP2D6 strips the methyl cap away and leaves behind a bare hydroxyl, an oxygen bonded to a single hydrogen. That single edit, the loss of 1 carbon and 2 hydrogens, a change almost too small to see, transforms Ibogaine into a new molecule. Its name is noribogaine, and noribogaine is the one that stays. This is the shape you should carry into the dark tonight, the refrain we will keep returning to, the parent drug for the experience, the metabolite for the durable reset. Ibogaine is the lightning strike, sudden, blinding, brief, gone almost as quickly as it came. Noribogaine is the slow afterglow, the warmth that lingers in the air long after the flash, the light that keeps the sky pale for a while after the storm has moved on. The visionary night, the waking dream, the flood of old memory, that is the lightning, and it fades within a day. But something in the body has been quietly reset, and that quieter work belongs to the afterglow. To understand why, you have to understand how long each of them lives. Pharmacology has a way of measuring the lifespan of a molecule inside the body, and it calls that measure the half-life, the time it takes for the concentration in the blood to fall by half, and then by half again, and then again, the slow geometric fade of a substance being cleared away. Ibogaine's half-life is measured in hours. A handful of hours and half of it is gone. Another handful and half of what remained is gone too. And so, within a day or two, the parent drug has almost entirely left. The lightning does not linger. It cannot. CYP2D6 is busy converting it. The kidneys and liver are busy clearing it. And the storm that seemed so total burns itself out with surprising speed. But noribogaine, the molecule left behind after the methyl cap is stripped, has a half-life measured not in hours, but in days. Days. It is cleared far more slowly, so it accumulates and it lingers, drifting through the bloodstream long after the parent has vanished, long after the visions have closed their eyes, long after the person has come back into the ordinary light of an ordinary room and thought the whole thing was over. It is not over. The afterglow is still there, dissolved in the blood, still touching receptors, still doing something to the wanting. And this, more than anything Ibogaine itself does in its brief, violent hours, may be the true workhorse of the anti-craving effect, the reason the relief can outlast the drug by weeks. So, consider the profile of this quieter molecule, because it is not simply a faded copy of its parent. Noribogaine binds where Ibogaine binds, but it presses on some targets harder and on others more gently, and the balance of its touch is its own. It reaches first for the kappa opioid receptor. This is one of the 3 great families of opioid receptors, mu, delta, and kappa. And the kappa receptor is the strange one, the dark one, the receptor whose full activation does not bring the warm euphoria of the mu, but rather dysphoria, unease, the grey weather of the mind. Noribogaine touches this receptor as a partial agonist, or perhaps as a modulator, pressing the key but never all the way down, turning the signal partly on without ever bringing the full storm of kappa activation. And this partial pressure at the kappa opioid receptor is one of the leading suspects in the mystery of craving, because the kappa system is woven deeply into the machinery of relapse, into the low pull that draws a person back toward the substance they were trying to leave. To touch it, gently, without overwhelming it, to hold it in a partial embrace for days on end, may be to hold the door of craving quietly shut. It reaches, too, for the mu opioid receptor, the same receptor morphine fills, the same receptor heroin floods, the same snug lock that opioids fit so perfectly. Noribogaine has activity here as well, weaker, more ambiguous, its exact character still argued over in the literature. But that it touches the mu receptor at all is part of why Ibogaine has drawn such interest for opioid dependence specifically. The notion that as the metabolite lingers, it may occupy or modulate the very receptors that withdrawal has left screaming, softening the crash, easing the body's long complaint. And it reaches still for the serotonin transporter, the SERT, the same pump we met in the parent drug, the little molecular ferry that clears serotonin out of the synapse and carries it home. Noribogaine continues to inhibit SERT, continues to block that reuptake, continues to leave serotonin lingering longer in the gap between neurons. And because noribogaine itself lingers for days, this serotonergic modulation is not the brief flicker of the acute experience, but a sustained, low, quiet pressure on the mood system, held steady across the very weeks in which the craving stays lifted. So tally it again, softly, the way you might count the lights of a distant town before sleep. Partial agonism at the kappa opioid receptor. Activity at the mu opioid receptor. Continued inhibition of the serotonin transporter. Not one snug fit, not one clean cause, but a small handful of gentle, simultaneous pressures, sustained for days by a metabolite that will not leave. This is the pharmacological reason craving may stay suppressed for weeks after a single dose. Not because the drug is still there, but because its afterglow is touching several systems at once, lightly, patiently, all at the same time. The lightning gives the experience. The afterglow gives the reset. Say it again in the dark. Let it settle. The parent drug for the experience, the metabolite for the durable reset. But there is a shadow in this, and you should meet it now, calmly, the way you meet any hard fact before sleep, without alarm, but without looking away. The shadow lives in that first enzyme, CYP2D6, the one that strips the methyl cap and turns lightning into afterglow. Because CYP2D6 is not the same enzyme in every person. The gene that builds it is one of the most variable genes in all of human pharmacology, a stretch of DNA that comes in dozens of forms, dozens of subtly different versions passed down through families and populations across the world. Some people carry versions that build a fast, tireless enzyme, and they are called ultra-rapid metabolizers. Some carry versions that build a slow, sluggish enzyme, or barely build a working enzyme at all, and they are called poor metabolizers. And most people fall somewhere in the wide country between. Think of it this way. The enzyme is the hand that converts the strike into its afterglow. In one person, that hand is quick. It seizes the Ibogaine and converts it rapidly to noribogaine, so the parent drug clears fast and the metabolite floods in early. In another person, that hand is slow, almost still. The Ibogaine lingers far longer than it should in its raw, unconverted form, building to concentrations the body was never meant to hold, while the gentle afterglow of noribogaine arrives late and thin. Same dose, same milligrams swallowed. Wildly, dangerously different journeys through the body. This is the CYP2D6 polymorphism, and it feeds two problems at once, two halves of the same uncertainty. It feeds the variance in whether the treatment works at all, because if the anti-craving effect belongs largely to noribogaine, then a person who converts poorly to noribogaine may get all the lightning and little of the afterglow, all the storm and none of the reset. And it feeds the variance in who lives and who dies, because the parent drug, Ibogaine, when it lingers at high concentration in a slow metabolizer, is the more cardiotoxic of the two, the one that reaches into the heart's electrical rhythm and stretches the interval between beats, the one that has stopped hearts. A poor metabolizer, given a standard dose, may be carrying a load of unconverted Ibogaine far beyond what a fast metabolizer would ever accumulate. And no one can see this by looking. Two people sit in the same room, swallow the same measured dose, and one moves smoothly toward the afterglow while the other drifts toward the edge of a lethal arrhythmia. And until the genotype is known, until the enzyme speed is measured, the difference is invisible. This is why the same substance can be, in one body, a night of hard visions followed by weeks of unexpected quiet, and in another body, a heart that falters and does not recover. Not because the drug changed, because the hand that converts it changed. Because CYP2D6 is not one enzyme, but 1,000 slightly different enzymes distributed unevenly across all of us, deciding, quietly, invisibly, before the person feels anything at all, how much lightning and how much afterglow each nervous system will receive. And so the mystery deepens rather than resolves, which is the honest shape of it. We now have two molecules where we thought we had one. We have a parent that burns bright and brief and delivers the visionary storm, and a metabolite that lingers for days and touches the kappa opioid receptor, the mu opioid receptor, and the serotonin transporter all at once, and seems to be where the durable reset lives. We have a single enzyme standing between them, and that enzyme differs from person to person in ways that shape both whether the medicine works and whether it kills. That is a great deal of knowledge. It is also, still, not an answer to the central question, the one we keep circling in the dark. When the wanting simply lifts and stays lifted for weeks, we still cannot point to a single place and say, there, that is the cause. But we can point, now, to a little more of the landscape. We can point to the lightning and name it Ibogaine, brief, cleared in hours, converted in the liver. We can point to the afterglow and name it noribogaine, lingering for days, pressing gently and simultaneously on three quiet systems of mood and craving and pain. We can point to the hand between them and name it CYP2D6 and acknowledge that its speed is written into our genes before we are born, and that this hidden variation is why the same night can be, for different people, a gentle reset or the last one. So let the image stay with you as the dark deepens, the strike and the glow, the flash that is over almost before you have registered it, and the pale, patient light it leaves behind, dissolved in the blood, working slowly on the wanting, long after the person believes the whole strange night is finished. The parent drug for the experience, the metabolite for the durable reset. One brief, one long. One loud, one quiet. Both present in their turn in a body that never once was asked which enzyme it had been given. A body simply doing, in the dark, what its own inherited chemistry has always quietly done. So we come now to the hardest and the most beautiful part of the story, the part where the science stops narrating and begins to guess. Because everything we have described so far, the receptors touched, the enzymes engaged, the metabolite that lingers, tells us what Ibogaine binds, but none of it tells us why the wanting quiets. A drug can occupy 100 receptors and change nothing that matters. The list of molecular contacts is not the same as the reason. And so the field has spent decades reaching for a single downstream event, one final common pathway into which all these scattered actions might drain, some deeper current beneath the surface chemistry that could explain the thing addicts keep reporting, that the craving, the old reflexive pull, is somehow gone. The leading candidate for that deeper current has a plain and unlovely name. GDNF, glial cell line-derived neurotrophic factor. Say it slowly, because it may be the most important molecule in this entire story, and almost no one outside a laboratory has ever heard of it. A neurotrophic factor is a protein the brain makes to feed, protect, and grow its own neurons, a kind of internal fertilizer for the wiring, a signal that says to a cell, stay alive, extend yourself, strengthen the connections you already have. GDNF is one of the most powerful of these, and it is especially potent for a particular population of neurons, the dopamine-producing cells clustered deep in the midbrain in a region called the ventral tegmental area, the VTA. Remember that name because it is the source of the river. From the VTA, dopamine neurons send their long, slender axons forward into a small structure near the base of the forebrain called the nucleus accumbens. This is the mesolimbic dopamine pathway, the reward circuit, the ancient machinery that evolution built to make food and water and sex and safety feel worth pursuing. It is not the pleasure of a thing, it is the wanting of a thing, the salience, the pull toward. And this is precisely the circuit that addiction captures. Every drug of dependence, however different its molecular entry point, the opioid at the mu receptor, the stimulant at the dopamine transporter, alcohol at the GABA A receptor, every one of them, by its own separate route, converges on this single stream, flooding the nucleus accumbens with dopamine, teaching the VTA over and over that the drug is the most important thing in the world. Addiction is, at bottom, a learning disorder written into this circuit. The habit is not a weakness of will. It is a groove carved by chemistry into living tissue. And here is the hypothesis in its simplest and most elegant form. When Ibogaine, or more likely its long-lived metabolite noribogaine, moves through the brain, it appears in rodents to make the neurons of the VTA produce more GDNF, more of their own fertilizer. And GDNF does something remarkable, something that turns a single push into a lasting change. It triggers a positive feedback loop. GDNF binds to its receptor complex on the dopamine neuron, a receptor called GFR-alpha-1, paired with a signaling partner called RET, a receptor tyrosine kinase that reaches through the cell membrane and lights up the machinery inside. And that machinery, once activated, tells the cell to make still more GDNF. the signal amplifies itself. A small initial rise becomes a self-sustaining wave of neurotrophic signaling that can persist long after the drug that started it has been cleared from the blood. One strike of the match and the fire keeps itself burning. Sit with the shape of that idea, because it is what makes the hypothesis feel like an answer. A single dose, a metabolite gone within days, and yet a downstream signal that, once kindled, feeds itself and outlasts its own cause. This is the molecular candidate for the thing patients describe, the sense that something durable was reset, that the change did not fade when the drug faded. And what does GDNF actually do inside those neurons once the loop is running? This is where the wanting might be rewritten. Neurotrophic signaling of this kind drives structural remodeling of the neuron itself, the growth of dendrites, the fine branching arms that receive signals, the strengthening or the pruning of synapses, the tiny junctions where one neuron speaks to the next. Addiction, over months and years, physically reshapes these connections in the reward pathway, thickening the synaptic architecture that makes the drug feel indispensable. The habit is not metaphorically carved into the brain. It is literally carved. In dendritic spines, in synaptic density, in the strength of specific connections between the VTA and the nucleus accumbens. And a surge of neurotrophic factor is one of the few things known to have the power to remodel that architecture, to loosen the carved groove, to make the circuit plastic again, malleable, capable of being rewritten. Not erased, remade. Every synapse the habit had strengthened, every dendrite it had thickened, every connection between the wanting source and the wanting center that years of use had reinforced, every circuit the habit had carved into the soft tissue of the self. GDNF, if the hypothesis holds, reaches into that architecture and makes it soft enough to change again. A reset. A return of plasticity to a circuit that had grown rigid around a single, overwhelming want. And notice, this is the part that makes the hypothesis so satisfying, how it gathers up all the scattered receptor actions we have spent this whole journey cataloguing and pulls them into one downstream stream. Remember them. The NMDA receptor, blocked. The kappa opioid receptor, engaged. The mu opioid receptor, weakly touched. The nicotinic receptors, antagonized. The serotonin transporter, inhibited. The sigma-2 receptor, bound. Each of these, on its own, seem to point in a different direction, a scattering of molecular gestures with no obvious common purpose. But if each of these actions, the NMDA blockade in particular, which is known in other contexts to influence synaptic plasticity and neurotrophic signaling, if each of them contributes, by its own small path, to the rise of GDNF in the VTA, then the scattering resolves. The disparate contacts become tributaries, and they all run downhill into the same neuroplastic river, the remodeling of the reward circuit. One drug, many locks, and beneath them all, a single deeper door. It is a beautiful hypothesis. I want you to feel how beautiful it is, drifting there in the dark, the elegance of a self-sustaining loop, the poetry of a groove made soft again, the way it ties a dozen loose threads into a single knot. And now I have to do the hard thing, the honest thing, the thing that separates a science story from a fairy tale. I have to tell you how much of this we do not actually know. Begin with the foundation. The strongest, cleanest evidence for the GDNF hypothesis comes from rodents, rats and mice. studies in which Ibogaine or noribogaine reduced self-administration of alcohol or other drugs, and in which that reduction was accompanied by measurable increases in GDNF in the ventral tegmental area, and, crucially, in which blocking GDNF signaling abolished the effect. That last detail matters enormously because it moves the finding past mere correlation towards something closer to cause. If you take the GDNF away, the anti-craving effect goes away too. In the rat, that is real, replicated, and mechanistically coherent. It is genuinely persuasive science. But a rat is not a human being. The rat brain is not the human brain scaled down. It is a different organ, with different densities of receptors, different metabolism, different circuitry laid over the same ancient reward pathway. A mechanism that is clean and causal in a rodent may be partial in a primate, or altered, or simply absent. The history of addiction pharmacology is a long graveyard of compounds that cured the rat and did nothing for the person. We do not have, in humans, the studies we would need to confirm this chain. The careful measurement of GDNF in the living human midbrain, the demonstration that Ibogaine raises it, the proof that the raising is what produces the durable change. We have anecdotes of interrupted addiction. We have a compelling animal mechanism. We do not yet have the bridge that firmly connects them. And so we have to be scrupulous about a distinction that is easy to blur when a story is this elegant. Correlation is not mechanism. That two things rise together, that Ibogaine is present and craving falls, that GDNF climbs and self-administration drops, does not prove that one caused the other, and it certainly does not prove how. The GDNF loop may be the whole answer. It may be one contributing thread among several. It may turn out to be a marker of something else entirely, a side effect that travels alongside the real mechanism without being it. We are looking at a footprint in soft ground and inferring the shape of the creature that made it. Sometimes the inference is right. Sometimes the ground was softer than we thought, and the print misleads. There are other candidate mechanisms too, quieter ones, that may act alongside or instead of the neurotrophic story. The interruption of opioid withdrawal itself, through those partial actions at the opioid receptors, giving the body a window of relief in which relapse becomes less inevitable. The dreamlike, memory-suffused, oniric state that many describe, a psychological reprocessing of the addiction that may do work of its own, independent of any single molecule. It is entirely possible that Ibogaine acts on several levels at once, biological and experiential braided together, and that no single hypothesis, however lovely, will ever hold the whole of it. The honest position is not that GDNF is wrong. The honest position is that GDNF is unproven, and that a mechanism can be both the best available explanation and still, in the end, incomplete. So let me restate the spine of this whole long night in its most distilled and unflinching form. We have a drug that appears to interrupt addiction. We have a plausible, elegant, partially supported hypothesis for how it might do so. A rise in GDNF, a self-sustaining loop of neurotrophic signaling, a remodeling of the mesolimbic reward circuit that softens the groove the habit had carved. And we do not, in humans, know that this is true. We have a beautiful hypothesis and we have no confirmation. That is not a failure of the story. That is the actual shape of the knowledge, a real thing observed at the bedside, a candidate mechanism traced in the rodent, and a gap between them that no one has yet crossed. Hold both of those things at once the way you can hold two temperatures of the same water. the wonder of a possible reset, a circuit made plastic again, a wanting loosened at its root, one dose reaching down into the architecture of desire and making it soft enough to change. And the discipline of not yet knowing. The refusal to call a hypothesis a fact simply because it is graceful, simply because we wish it were so. Both are true. The promise is real, the proof is not here. And still, imagine it, just as a picture to carry down into sleep. Imagine the ventral tegmental area, deep in the midbrain, its dopamine neurons quiet in the dark. Imagine a molecule slipping past them and leaving behind not a command, but an invitation. Make more of your own fertilizer, feed yourselves, grow. Imagine the loop catching, the signal beginning to amplify itself, RET lighting up inside the membrane, the message spreading cell to cell long after the drug is gone. And imagine, over the hours and days that follow, the slow, structural answer to that signal. Every synapse the habit had strengthened, every dendrite it had thickened, every connection it had reinforced, every circuit it had carved into the living tissue of a person's wanting, all of it becoming, for a little while, soft again, capable of being rewritten. That is the hypothesis. That is the beautiful, unproven thing. We know the match was struck. We know the glow it left behind. We can even name the fertilizer it may have called up out of the neurons themselves. What we cannot yet say, what no honest scientist will say, is that we have watched, in a human brain, the groove of an addiction fill in and smooth over and heal. We have a candidate. We have a mechanism drawn in the rat and sketched faintly into the person. And we have the quiet, patient, unfinished work of finding out whether the most hopeful hypothesis in the pharmacology of addiction is, in the end, actually true. So let us leave the microscope for a while and step inside a single human day, though calling it a day undersells it, because what unfolds when a person swallows a full flood dose of Ibogaine is closer to a day and a half, roughly 36 hours of altered time, arranged in phases as orderly as the tide, each phase married not to spirit, but to the slow, measurable rise and fall of a molecule in the blood. Imagine the day unfolding behind your closed eyes. You have taken the capsule, Ibogaine hydrochloride, a purified salt, or perhaps the ragged total alkaloid extract of the iboga root, and already the pharmacokinetics have begun their quiet arithmetic. Ibogaine is absorbed through the gut wall and swept first to the liver, where the enzyme CYP2D6 waits to convert it. This is the first-pass metabolism we spoke of, and within the first hour or two, the parent compound, Ibogaine itself, climbs toward its peak concentration in the plasma. That peak is the opening curtain. That peak is phase 1, and phase 1 is the flood. This is the oniric state, the waking dream, the hours most often mistaken for a visitation from somewhere beyond the self. With your eyes closed, you see landscapes assemble and dissolve, corridors of imagery, faces, geometry, long unspooling narratives that feel watched rather than authored. Dreams, in other words, running while you remain awake enough to know that you are lying still in a darkened room. But nothing here has stepped in from outside. What you are experiencing is the direct signature of a molecule sitting on several of the brain's most important locks at once and turning them all the wrong way. Consider what Ibogaine is touching at this peak. It binds, with modest affinity, to the serotonin transporter, the SERT, the same protein the SSRIs occupy, and by interfering with the reuptake of serotonin, it lets that neurotransmitter linger in the synaptic cleft, tilting the serotonergic tone of the whole forebrain. It reaches, too, toward the serotonin 2A receptor, the 5-HT2A site, the very receptor through which classical psychedelics like psilocin and LSD generate their visions. Ibogaine's grip there is looser, more ambiguous, but present, and it may be part of why the imagery comes. And most importantly, most distinctively, Ibogaine is sitting inside the NMDA receptor, the glutamate-gated ion channel that governs how neurons learn, wedged as an open-channel blocker deep in the pore where the dissociatives ketamine and PCP also lodge. Block the NMDA receptor, and you loosen the ordinary binding of perception to reality. You let the brain's internal model drift free of its usual correction. Serotonin disturbed, glutamate damned. That is the pharmacology of the waking dream, and it is enough, more than enough, to paint the corridors behind your eyes without a single ghost being required to explain them. And here we must be honest, gently but firmly honest, the way we have tried to be honest all along. People come out of phase 1 describing meetings with ancestors, with a stern maternal presence, with the architecture of their own childhood rebuilt around them in the dark. These reports are real as experiences. They are not evidence of mechanism. The subjective texture of a state and the molecular cause of a state are two different registers, and the fact that a visionary hour feels meaningful tells us nothing, by itself, about what the drug is doing to the receptors. We can say that serotonin is elevated and NMDA is blocked. We cannot say the brain has spoken with anyone. Hold both of those truths at once, the vividness and the uncertainty, and you are holding this drug correctly. Phase 1 lasts, for most, something like 4 to 8 hours, riding the plateau of peak parent ibogaine in the blood, and then, as the liver keeps working and the plasma concentration of the parent compound begins its descent, the character of the experience changes with it. The tide turns, the flood recedes, and phase 2 begins. Phase 2 is quieter, and stranger for being quiet. The rushing imagery thins. The corridors stop assembling themselves, and in their place comes a long, sober, evaluative stillness, an introspective phase in which many people report the peculiar phenomenon that has given Ibogaine so much of its reputation: the life review. Memory after memory surfaces, not as dream, but as recollection, often autobiographical, often uncomfortable, often organized around the very behavior that brought the person to the drug in the first place. Old scenes are re-examined with a flat, almost clinical detachment, as though the emotional charge has been temporarily unhooked from the content, and you are permitted to look at your own history without flinching from it. And it is tempting, it is deeply tempting, to reach for the mystical here, to say the medicine is showing you your life. But stay with the molecule. In this phase, the parent ibogaine is falling, while its principal metabolite, noribogaine, produced when CYP2D6 strips a methyl group from the parent, is rising to take its place. Noribogaine has its own pharmacology, overlapping but not identical. It, too, blocks the serotonin transporter, and it engages the opioid system, showing activity at the mu and kappa opioid receptors that the parent compound engages more weakly. So the serotonergic disturbance persists, even deepens, while the raw psychedelic push of the parent compound wanes. And it may be precisely that combination, high serotonergic tone without the overwhelming visionary flood, that opens the calm, reflective, emotionally flattened window in which the life review unfolds. Maybe. We say maybe because the honest map here is still sketched in pencil. What people do in that window, the reappraisal, the resolve, the sense of having settled something, is subjective report. And subjective report, however consistent, is not mechanism. Still, notice how the phases track the chemistry. Phase 1 rides the parent compound at its peak, all serotonin and blocked glutamate and unspooling vision. Phase 2 rides the crossover, the parent falling and the metabolite climbing, vision giving way to reflection. The experience does not float free of the body. The experience is the body's chemistry, read from the inside, the plasma curve translated into hours of the mind. And that is the quiet thesis we keep returning to, the drug that interrupts addiction and nobody knows how. We can name the molecules, we can time the phases against their rise and fall, and we still cannot fully close the gap between the concentration in the blood and the meaning in the mind. Then comes phase 3, and phase 3 is the long tail, the part the mythology forgets because it is not beautiful. The visions are gone, the life review has closed, but the ordeal is not over because the pharmacokinetics are not over. This is where the second signature molecule of the whole story asserts itself completely, because noribogaine has a remarkably long half-life, far longer than the parent compound, persisting in the plasma not for hours, but for days, elimination stretching out across a slow, extended decline. And so, even as the acute drama fades, the drug is still present, still occupying the serotonin transporter, still keeping serotonergic tone elevated, and the body registers that persistence as a long, residual stimulation. Sleep does not come. This is the insomnia phase, and it is one of the most reliably reported features of the entire experience. A wired, restless, exhausted wakefulness that can stretch across the second night and into the days beyond. The mind quiet at last, but the body refusing to power down. Picture it. The flood has passed. The review is finished. You are spent in every way a person can be spent, and still you lie awake, because a long-lived metabolite is sitting on your serotonin transporters and will not step off. The stimulation is not spiritual restlessness. The stimulation is a pharmacokinetic fact. Noribogaine's long half-life, translated into hours of a body that cannot yet sleep. And here, the danger threads quietly back into the story, because phase 3 is not merely uncomfortable. It is the window in which the cardiac risk we have circled before does its patient work. Both Ibogaine and noribogaine block the hERG potassium channel, the ion channel that governs the heart's electrical recovery, the current that lets each beat reset itself for the next. And when that channel is blocked, the interval called QT lengthens, the reset slows, and the heart becomes vulnerable to the chaotic rhythm that can kill. Because noribogaine lingers so long, that vulnerability lingers too, out past the visions, out past the insight, into the days of residual wakefulness when everyone has assumed the worst is over. This is why the honest clinics keep the cardiac monitor running long after the last vision has faded, not because the mind is still in danger, but because the metabolite is still in the blood, and the blood is still touching the heart. So imagine the whole arc now, the full 36 hours and their long, fraying tail, laid out as a single, unbroken curve. the capsule swallowed, the gut absorbing, the liver's CYP2D6 taking its first pass, the parent compound rising to its peak, and, at that peak, the flood. Serotonin loosed, NMDA channels dammed, 5-HT2A brushed, the waking dream painting its corridors behind your closed eyes for 4 hours, 6 hours, 8. Then the turn, the parent falling and noribogaine rising, the crossover hours, the visions thinning into the flat, clear light of the life review, memory after memory examined without flinching. Then the long descent, the parent nearly gone, the metabolite reigning alone, its long half-life stretching the drug's presence across days, keeping the serotonin transporter occupied, keeping the body wired and wakeful, and, quietly, invisibly, keeping the hERG channel blocked and the heart's reset slowed. 1 molecule, 2 forms, 3 phases, 36 hours and more, each part of the experience shadowing a measurable curve in the plasma. That is the shape of the thing from the inside. That is what a person means when they say they have gone through Ibogaine. Not a single event, but a sequence. A tide with a rising limb and a falling limb and a long, slow ebb. Each stage the direct expression of how much parent compound and how much metabolite happen to be circulating at that hour. And we can chart it, honestly and precisely. We can name every receptor the molecule leans on as it rises, and every channel it lingers on as it falls. And yet the deepest question stays open, the same question it has been from the beginning. We know the corridors were painted by serotonin and glutamate. We do not know whether the life review, whatever it truly is doing any of the healing. We know the body was flooded and then wired. We do not know how much of the interruption of addiction happens during these 36 hours, and how much happens quietly afterward, in the days and weeks when the neurotrophic signals we spoke of earlier may still be at work, long after the last of the noribogaine has finally cleared the blood. Hold both truths one more time and let them settle. The experience is real, phase by phase, hour by hour, as real as anything a body can undergo. And the meaning of the experience, whether the visions matter, whether the review heals, whether any of the felt drama is the mechanism or merely the weather that accompanies it, remains for now the beautiful, unproven thing at the center of it all. The drug that interrupts addiction, and nobody yet knows how. There is another truth we have circled all this time, and we can no longer walk around it. It is the darkest fact in this whole quiet story, and it deserves to be told plainly, without softening, without the comfort of metaphor, unless the metaphor helps you understand. The same molecule that resets the mind can stop the heart. Not metaphorically, not rarely enough to ignore. Ibogaine kills, and it kills through a mechanism that pharmacology understands with painful precision, even as the mechanism of its healing remains a mystery. So let us hold this last, heaviest truth the way we have held all the others, slowly, carefully, and with our eyes open. To understand how Ibogaine stops a heart, you must first understand how a heart keeps its rhythm at all. Every beat of the heart is an electrical event. A wave of voltage sweeps across the muscle, cells depolarizing in sequence. The interior of each cell flooding briefly positive as sodium and calcium ions rush inward through their channels. That is the contraction, the squeeze that pushes blood outward. But a contraction is only half a heartbeat. After the squeeze must come the release, the repolarization, the return of the cell to its resting negative charge so that it can be ready to fire again. And that return is carried, more than anything else, by potassium. Potassium ions flowing outward through their channels, draining the positive charge back out, resetting the voltage, ending each beat cleanly so the next one can begin. There is one channel that matters most for this reset, and it has a name you should remember, because Ibogaine's shadow lives inside it. It is called hERG, the human ether-agogo-related gene, encoded by the gene KCNH2, and it carries a current cardiologists call IKR, the rapid delayed rectifier potassium current. Picture it as a floodgate in the wall of every heart cell, a gate that opens late in each beat and lets potassium pour out fast so that repolarization arrives on time and the rhythm stays sharp and regular. Every heartbeat depends on this gate. Every clean interval between beats is IKR doing its quiet, unglamorous work, closing the door on one contraction so the next can open. And Ibogaine binds to this gate and blocks it. That is the whole of the danger in a single sentence. Ibogaine is a potent blocker of the hERG potassium channel. It slides into the channel and obstructs the flow of potassium. And when potassium cannot leave the cell quickly enough, repolarization slows. The reset comes late. The electrical event of each heartbeat stretches out longer than it should, and that stretching shows up on an electrocardiogram as a lengthened interval. The QT interval, the span from the start of the ventricular squeeze to the end of its recovery. Ibogaine prolongs the QT interval. In case after case, in dose after dose, the trace on the monitor grows visibly, dangerously long, and every heartbeat, every interval, every fragile rhythm the drug can stretch too far becomes a small delay accumulating toward disaster. Why does a stretched interval matter? Because a heart cell that has not finished repolarizing is a heart cell that can misfire. When repolarization drags, the recovering cell can throw off a small premature spark, an early after depolarization, a stray voltage that arrives before the cell was ready, at exactly the wrong moment in the cycle. And in a heart where every cell is repolarizing late and unevenly, one stray spark can catch and spread and tip the whole coordinated wave into chaos. This is the arrhythmia that Ibogaine can cause, and it has a name as beautiful as it is deadly: torsade de pointe, the twisting of the points. On the electrocardiogram, the peaks appear to rotate around the baseline, twisting like a ribbon caught in wind, and the heart, no longer beating, only quivers. Torsade de pointe can resolve on its own. It can also degenerate into ventricular fibrillation, the terminal rhythm, the heart trembling uselessly while the blood stops moving. That is how Ibogaine kills. Not through the visions, not through the flood, through the silence of a potassium gate held shut too long. Now recall what we learned about how the body handles this molecule, because the pharmacokinetics do not soften the danger, they sharpen it. Ibogaine is metabolized in the liver by the enzyme CYP2D6, the cytochrome that strips a methyl group and converts ibogaine into its long-lived metabolite, noribogaine. And here, two facts we have already met return with a colder meaning. First, CYP2D6 is genetically variable. A meaningful fraction of people are poor metabolizers. Their CYP2D6 works slowly or barely at all. And in those bodies, Ibogaine itself lingers at high concentration far longer than expected, keeping the hERG blockade in force, keeping the QT interval stretched, hour after hour, with no way for the person or the underground guide to know their genotype in advance. Second, noribogaine, the metabolite, the one with the very long half-life we described, measured not in hours, but in days. Noribogaine also blocks hERG. So even as the parent drug clears, the metabolite persists, and the cardiac risk does not end when the visions fade or when the acute phase passes. The danger window stretches across days. Every hour that noribogaine remains in the plasma is an hour in which the potassium gate stays partly shut and the rhythm stays vulnerable. The long half-life that some hope prolongs the healing also, unmistakably, prolongs the risk of death. And the heart's vulnerability is not fixed. It depends on the sea the cells are bathed in, the concentrations of ions in the blood. And here, two electrolytes matter enormously. Potassium, in the blood, sets the baseline for how easily potassium can leave the cell. When blood potassium is low, hypokalemia, the outward current weakens, repolarization slows further, and the QT interval lengthens even before a single molecule of ibogaine arrives. Add the hERG blockade on top of already low potassium, and you have stacked one delay upon another. And magnesium. Magnesium is the ion that stabilizes the cardiac membrane, that suppresses those stray early after depolarizations, that guards against torsades directly. Low magnesium, hypomagnesemia, removes that guard. It is no accident that the emergency treatment for torsade de pointe is intravenous magnesium. It is the ion that calms the twisting rhythm. But in someone with low magnesium and low potassium taking ibogaine, every protective factor is absent, and every provocative factor is present at once. And who arrives at these ceremonies with depleted electrolytes? Very often, the very people ibogaine is meant to help. People in withdrawal. People who have been vomiting, not eating, dehydrated, their bodies stripped of magnesium and potassium by weeks or months of opioid or alcohol dependence and the sickness of coming off it. The population most drawn to ibogaine is, too often, the population least electrically prepared to survive it. This is where the story turns from mechanism to consequence, and the consequences are not hypothetical. There are documented deaths, real people, named in case reports and coroner's findings, who took ibogaine and died of cardiac arrest, of fatal ventricular arrhythmia, of the sequence we have just traced. Reviews of these deaths find the fingerprints again and again. Prolonged QT, torsades, a heart that stopped in the hours or days after the dose. Many of the dead had pre-existing conditions, a heart already scarred, a rhythm already fragile, a congenital long QT the person never knew they carried, a history of arrhythmia, structural heart disease, or the use of other medications that also block hERG or deplete electrolytes. Some had taken methadone, itself a QT-prolonging drug, layering blockade upon blockade. And nearly all of them took ibogaine in the setting that makes every one of these dangers worse, the underground. Let us be precise about what underground means here, because the setting is not incidental. It is often the difference between a stretched interval that resolves and a heart that never restarts. In a properly equipped cardiac setting, ibogaine's dangers are, at least in principle, manageable. You would screen the heart first with an electrocardiogram and refuse anyone with a baseline QT already long. You would measure serum potassium and magnesium and correct them to safe levels before a single dose. You would place the person on continuous cardiac telemetry, a monitor watching every beat, every interval, in real time, so that a lengthening QT could be caught as it happened and the dose stopped. You would have a defibrillator in the room and intravenous magnesium drawn up and ready, and staff trained to recognize torsades in its first twisting seconds and to shock a fibrillating heart back into rhythm. Every one of those safeguards exists because the mechanism is understood. And in the underground, almost none of them are present. No electrocardiogram, no electrolyte panel, no telemetry watching through the long night, no defibrillator, often no medical training at all, only a guide and hope, and a person lying in the dark while a molecule quietly stretches every interval of their heartbeat toward the edge, with no one measuring how close to the edge it has come. So hold, once more, the terrible symmetry at the center of this drug. The very features that make Ibogaine pharmacologically extraordinary are inseparable from the features that make it lethal. The long half-life of noribogaine that keeps the neurotrophic signals humming for days is the same long half-life that keeps the hERG channel blocked for days. The broad, promiscuous binding across so many receptor systems, the NMDA channel, the kappa and mu opioid receptors, the sigma sites, the serotonin transporter, that same lack of selectivity means the molecule does not politely avoid the potassium channel of the heart. It cannot be tidied. The healing and the harm are not two separate drugs. They are two faces of one molecule, and there is no dose yet known that reliably delivers the first without risking the second. The same molecule that resets the mind can stop the heart. Say it plainly, believe it fully. The same molecule that resets the mind can stop the heart. And this is why the epistemic honesty we have practiced all along must, in this one chapter, refuse any comfort. Elsewhere we have said we do not fully understand the healing and left the mystery open with a kind of wonder. Here there is no wonder. There is only clarity, and the clarity is grim. We understand the killing far better than the curing. We can name the gene, KCNH2. We can name the channel, hERG. We can name the current it carries, IKR, and the interval it protects, the QT, and the arrhythmia that follows when the interval grows too long, torsades de pointe, and the terminal rhythm it decays into, ventricular fibrillation. We can name the enzyme that governs exposure, CYP2D6, and the poor metabolizers whose bodies cannot clear the drug, and the long-lived metabolite, noribogaine, that carries the danger onward across days. We can name the ions whose absence removes the last defenses, potassium and magnesium. The science of how Ibogaine ends a life is not a mystery at all. It is a chain of well-lit, well-documented steps, each one following the last with the cold reliability of physics. It is the science of how it saves a life that remains dark. Lie still and feel your own heartbeat for a moment, not with fear, only with attention. Feel the steady return of it, beat after beat, each one squeezing and releasing, each one depending on that quiet, outward flow of potassium, closing the door on the last contraction so the next can begin. You are, right now, a rhythm sustained by a floodgate you never think about, an interval kept clean by a channel you cannot feel. Most nights, that gate opens and closes millions of times without a single error, a fidelity so perfect it becomes invisible. Every heartbeat, every interval, every fragile rhythm, sustained tonight by nothing more dramatic than potassium leaving a cell on time. And Ibogaine is the molecule that can make it leave too late. This is not a warning meant to frighten you out of sleep. It is the counterweight the whole story requires, the dark mass on the other side of the scale, without which nothing we have said about hope and healing means anything honest at all. A drug that could not hurt you would be easy to speak about. Ibogaine is not easy. It offers, in one hand, a way out of dependence that almost nothing else in medicine can offer, and in the other, a lengthened QT interval and a heart that may lose its rhythm in the dark. Both hands belong to the same molecule. And so, the story of Ibogaine was never going to be a simple one, and the people who governed it were never going to find it easy either. To understand how a compound this strange came to sit where it sits, we have to travel back to a particular year and a particular act of law and watch a door close that has not fully opened since. The year is 1970. In the United States, the Controlled Substances Act is passed, and with it comes a system of schedules, a set of drawers into which every psychoactive molecule must be sorted. Schedule 1 is the deepest drawer, the one reserved for substances declared to have a high potential for abuse and, in the exact language of the statute, no currently accepted medical use. Into that drawer, alongside heroin and LSD and cannabis, Ibogaine was placed. The classification was not a judgment about Ibogaine's mechanism because almost no one understood its mechanism. It was a judgment about its family, a hallucinogen, a psychedelic, a thing that altered the mind. And in the climate of that year, that was enough to close the drawer and turn the key. Consider the strangeness of the placement. Schedule 1 is defined, in part, by that phrase, no currently accepted medical use. And Ibogaine, at that very moment, was a molecule whose most remarkable property was arguably medical. The interruption of opioid withdrawal, the quieting of craving, the reset of a dependent brain. The law was describing the one thing the drug might be best at as the very thing it did not possess. It was as if the statute had reached for the molecule's single greatest gift and written in the margin, none. And the mechanism the law ignored was not trivial. By the time Ibogaine entered that deepest drawer, its reach across the nervous system was already in fragments becoming visible. This was a compound that touched the mu opioid receptor, the very site where morphine and heroin and oxycodone dock, and where dependence is built, molecule by molecule, over months of use. It was a compound whose primary metabolite, noribogaine, the longer-lived shadow that CYP2D6 carves from the parent drug and sends circulating for days, behaved as a modulator at that same opioid site, and as a blocker of serotonin reuptake, holding serotonin in the synapse the way certain antidepressants do. It was a compound that antagonized the NMDA receptor, the glutamate-gated channel through which the nervous system encodes tolerance and learns chemically to need more. It was a compound that touched nicotinic acetylcholine receptors, the alpha-3 beta-4 subtype clustered in the habenula, a small, deep brain structure increasingly implicated in the machinery of aversion and withdrawal. One molecule, many locks. and the law had sorted it by its silhouette, not its keys. So the drawer closed. And in the United States, for decades, it stayed closed. The scheduling did not merely restrict Ibogaine's sale, it strangled its study. To research a Schedule I compound is to enter a bureaucratic labyrinth of federal licenses and registrations, of secured storage and monitored supply, of grant committees reluctant to fund work on a molecule the government has already declared worthless and dangerous in the same breath. The paradox compounds itself here, quietly and cruelly. A drug is placed in Schedule I partly because it has no accepted medical use, and the scheduling then makes it nearly impossible to perform the very research that could establish a medical use. The drawer locks from the inside. The molecule cannot prove itself useful because it has been declared, by law, unstudyable in any ordinary way. This is not a flaw unique to Ibogaine. It is the structural trap of Schedule I. But few molecules had as much to lose in that trap as this one. And what makes the loss sting is the timing. For consider what was unfolding in the same country, across the same decades, while Ibogaine sat in its drawer. The opioid crisis did not arrive all at once, but it arrived. First as prescription pain medication distributed with a confidence that would later curdle into scandal. Then as heroin filling the gap when the prescriptions were cut off. Then as fentanyl and its analogues, molecules so potent that a quantity smaller than a snowflake can stop a person's breathing. Dependence spread through the mu opioid receptor the way it always has. Repeated agonism, the receptor's own adaptive downregulation, the nervous system recalibrating around the constant presence of the drug until its absence becomes a physiological emergency. Tolerance climbing, doses climbing. The habenula and the locus ceruleus lighting up in withdrawal. Hundreds of thousands of deaths accumulating year over year, a toll that would eventually be counted in the millions of years of life lost. And through all of it, in the deepest drawer of the schedule, sat a molecule whose signature property, the one the law had marked as none, was the interruption of exactly this. A compound that could, in a single extended session, blunt the withdrawal that drives so much relapse, quiet the craving that outlasts the detox, and hand a dependent brain something that felt, to those who received it, like a reset. Not a cure, never a cure, but an interruption, a window, a door briefly held open in a disease defined by doors slamming shut. The country had a catastrophe of opioid dependence and, filed away under no accepted medical use, one of the few molecules that seemed to reach directly into the machinery of that dependence and loosen its grip. The irony is almost too heavy to hold. It is the kind of irony that does not announce itself loudly. It simply sits there, year after year, in the quiet arithmetic of the overdose statistics and the locked laboratory doors. But molecules do not stay buried simply because a statute wishes them to. Demand does not respect a schedule. And so, as the American door stayed shut, another response took shape. Not in universities or hospitals, but at the edges, across borders, in the places where the law's writ ran thinner. If Ibogaine could not be studied or offered inside the United States, then those who wanted it, and those who believed in it, would go to where it could. They went south, mostly. To Mexico, where clinics opened in the coastal cities and the desert towns, offering Ibogaine treatment in a space that was neither fully legal nor clearly forbidden. A legal grey zone, unscheduled and unregulated, where the molecule occupied no drawer at all. To Costa Rica, and to other countries scattered across the map, wherever the regulatory silence left room. An entire treatment culture grew up in this borderland, part medicine, part refuge, part improvisation. People who had exhausted every sanctioned option, the methadone, the buprenorphine, the abstinence programs, the relapses stacked one atop another, crossed borders to sit in a room and swallow a capsule of a molecule their own country had declared to have nothing to offer them. And here the two halves of the story, the promise and the peril, come together in the most dangerous possible way. Because the underground is exactly the setting in which Ibogaine's darkest property becomes lethal. Recall what the molecule does to the heart, how it blocks the hERG potassium channel, the delayed rectifier through which potassium must flow to end each heartbeat cleanly, how that blockade lengthens the QT interval, how a lengthened QT interval can slide, without warning, into the chaotic spiral of Torsades de Pointe. Recall that this danger is not random. It is worsened by low potassium, by low magnesium, by pre-existing cardiac conditions, by the simultaneous presence of other QT-prolonging drugs, by the very opioids and their metabolites that may still be circulating in a body that has only just stopped using. Recall that the treatment session itself, the long dose, the sustained high concentrations of Ibogaine and its lingering metabolite, noribogaine, is precisely the window in which the rhythm is most likely to fail. Now place that session in a clinic operating outside regulation. A clinic that may or may not measure a patient's serum potassium and magnesium before dosing. That may or may not run an electrocardiogram to see the QT interval before it is stretched, and again during the session to watch it stretch. That may or may not have a cardiac monitor beeping steadily through the night, a defibrillator within reach, staff trained to recognize the twisting of the points on the trace and to act in the minutes that matter. Some of these clinics do all of this, carefully and well. Some do almost none of it, and the patient, arriving desperate, is rarely in a position to tell the difference. The deaths that have been recorded in association with Ibogaine cluster heavily here, not in the molecule's mechanism alone, but in the collision between that mechanism and the absence of the monitoring that could catch it. The hERG channel does not care where it is blocked. It fails in a well-equipped hospital and in a beachside clinic the same way. The difference is only whether anyone is watching the trace when it does. So we arrive at the strange and unhappy equilibrium the story has been building toward, and it is worth laying the pieces out plainly, one beside the other, the way you would lay out the contents of a drawer you were finally forced to open. There is real promise, a molecule that reaches into the mu opioid receptor, antagonizes the NMDA channel, modulates the alpha-3 beta-4 nicotinic receptor in the habenula, and, through some combination we still cannot fully name, interrupts the cycle of dependence in a way almost nothing else can. There is real lethality, the hERG blockade, the lengthened QT, the possibility of a heart losing its rhythm in the quiet of the treatment night. There is no legal framework, the Schedule I classification that forbids ordinary study and sanctioned care inside the country where the need is greatest, and the regulatory silence beyond its borders that permits treatment without guaranteeing safety. And there are desperate patients, people for whom the sanctioned options have failed, who will cross any border and accept any risk for the interruption this molecule offers. Real promise, real lethality, no legal framework, desperate patience. 4 facts that do not resolve into each other, that simply coexist, pressing against one another in the dark. This is the shape of the Ibogaine problem, and it has held this shape, more or less unchanged, for the better part of 50 years. The molecule that could not hurt you would be easy to schedule and easy to offer. Ibogaine is not that molecule. It offers the interruption in one hand and the arrhythmia in the other, and the law, faced with both hands, chose simply to lock the drawer and look away, which did not make either hand disappear. It only moved them somewhere darker, somewhere with fewer monitors and thinner oversight, somewhere the people most in need would have to go alone. And yet the molecule persists, as molecules do, indifferent to the schedules we assign them. Somewhere tonight, in a room across a border, a capsule is dissolving in a stomach, crossing into a bloodstream, beginning its slow first-pass conversion by CYP2D6 into the long-lived noribogaine that will circulate for days. Somewhere, a heart is beating its ordinary rhythm, potassium leaving each cell on time, unaware that the molecule now entering it will reach for that timing and slow it. Somewhere, a person who has tried everything is lying still, waiting for the interruption they crossed a border to find, hoping the hands that offer it are also the hands that will watch the trace. The promise and the peril, in the same room, in the same body, at the same hour. This is where the law's silence delivered us, not to safety and not to ruin, but to this uneasy, unmonitored middle, a molecule doing extraordinary things in the dark, with no one required, by any statute anywhere, to be certain that someone is watching. And then, quietly, someone began to watch again. Not in a hotel room across a border, but in a hospital, with a monitor running and a plan written down in advance. That is the shape of the modern revival, and it begins not with the addicted at all, but with the wounded. At Stanford, a psychiatrist named Nolan Williams gathered a small group of special operations veterans, men who had carried, in the tissue of their skulls, the invisible sediment of blast exposure and repeated concussion. Traumatic brain injury, layered with post-traumatic stress, depression, and the flat grey weight of lives that had stopped moving forward. These were not people looking for a party. They were people looking for a door. And what the Stanford team offered them was Ibogaine, the same alkaloid we have followed through the mu opioid receptor, through the NMDA channel, through the sigma-2 and the serotonin transporter, but offered this time inside a structure built to keep them alive. Because we know what Ibogaine does to the heart, we have said it and we will say it again, because it is the whole reason this molecule has lived in shadow. Ibogaine and its longer-lived metabolite, noribogaine, block a potassium channel in cardiac muscle, the one encoded by the hERG gene, the channel that carries the rapid delayed rectifier current called IKR. That current is the outgoing tide that repolarizes each heartbeat, that pulls the cell's voltage back down and readies it to fire again. Block that channel and the tide comes in slow. The repolarization lags. On the electrocardiogram, the QT interval stretches. And in that stretched interval lies the danger, the disorganized, spiraling rhythm called torsade de pointe, the twisting of the points, the arrhythmia that has killed in Ibogaine's own history. So the Stanford team did something pharmacologically elegant. They gave magnesium. Consider what that means at the level of the ion. The heart's rhythm is a negotiation among charged atoms crossing membranes. Sodium in, calcium in, potassium out. Each current a voice in a chord. When hERG is blocked and repolarization drags, the myocardial cell becomes electrically unstable, prone to a stutter called an early after depolarization, a small, unwanted second spike that arrives before the cell has fully rested. That stutter is the seed of torsades. And magnesium, the humble ion, the fourth most abundant cation in the body, quiets exactly that seed. It stabilizes the myocardial membrane. It modulates the calcium currents whose untimely reopening drives the early after depolarization. Magnesium sulfate is, in fact, the standard emergency treatment for torsades already, the drug a cardiologist reaches for when the twisting begins. The Stanford approach simply moved it forward in time. Not a rescue after the arrhythmia, but a shield placed before it. Preload the blood with magnesium, keep the level high across the hours when Ibogaine and noribogaine are doing their slow work on hERG, and you blunt the very instability the potassium block creates. You cannot unblock the channel. The alkaloid will sit in it as long as it lingers, but you can make the surrounding tissue less willing to misfire while it does. The peril and the countermeasure, given together in the same vein, in the same hour. And the results, published in Nature Medicine in the year 2024, were striking. The veterans improved, measurably and across the map. Post-traumatic stress symptoms fell. Depression lifted. Measures of disability and daily functioning climbed towards something the men had not felt in years. Some described it in language that belongs less to pharmacology than to pilgrimage. A life returned, a self recovered. No serious cardiac events were reported under the magnesium protocol. It was, by every gentle measure, a beautiful result. And here we must slow down and be honest, because honesty is the only thing that keeps awe from curdling into hype. That study was small, a few dozen people. It was open label, everyone knew what they were receiving, veterans and researchers alike, and that knowledge alone can lift a mood, can bend a questionnaire, can conjure improvement out of hope. There was no placebo group, no blinded comparison, no way yet to separate the molecule's true effect from the enormous force of expectation and ceremony and the simple fact of being cared for. Promise is not proof. It was never proof. It is a signal, a bright, tantalizing, carefully collected signal. And a signal is an invitation to the harder work, not a conclusion. Every honest voice in that study said so themselves. But the signal was bright enough to be seen far beyond one hospital. Because behind all of this, behind every hopeful veteran and every cautious scientist, there runs a tide. And this tide is made of fentanyl. Here is the thing that makes a one-dose interrupter so unbearably tantalizing. The opioid crisis has changed its shape. It is no longer principally heroin, no longer principally the diverted prescription pill. It is fentanyl, a synthetic mu-opioid agonist so potent that it is dosed in micrograms, so lipophilic that it crosses into the brain almost instantly, so lethal in its narrow margin that a few grains too many stop the breath. Fentanyl binds the same mu-opioid receptor we have circled again and again, the receptor coupled to its inhibitory G protein, the receptor that closes the door on the drive to breathe. And it kills faster, more indiscriminately, and in greater numbers than any opioid in human history. Tens of thousands of people a year in one country alone. Against a tide like that, the idea of a molecule that could reset the craving in a single night, that could reach into the reward circuitry, upregulate the glial-derived neurotrophic factor called GDNF, quiet the dopamine transporter's frantic loop, blunt the withdrawal the NMDA channel amplifies, and let a person walk out the door without the pull that has ruled their life. The idea of that is almost unbearably attractive. One dose, one interruption, against a killer measured in micrograms. That attraction is now moving through the machinery of governments and money. In Kentucky, a state gutted by opioids, a state that buried a generation, there arose a debate over the settlement funds, the enormous pool of money wrung from the pharmaceutical companies that seeded the crisis. Should some of that money, the argument went, be directed toward Ibogaine research, toward trials that might finally answer, under proper controls, the question the underground has been asking for 50 years. It was a fierce and unresolved debate, and it stalled. But the very fact that it was had, that opioid settlement dollars were seriously proposed for a Schedule I hallucinogen, marks how far the conversation has travelled from the shadows. And then, in the year 2025, Texas moved. Texas, not a place one associates with psychedelic frontier science, committed public funding toward Ibogaine clinical trials, one of the largest such commitments a government has ever made toward this molecule. The stated goal was exactly the goal the science demands: proper trials, controlled trials, trials that might carry Ibogaine, if it survives them, toward the possibility of approval. Toward a world in which the molecule could be given not in a hotel room across a border, with no one required to watch the trace, but in a clinic, with magnesium in the vein and a cardiologist reading the QT interval, and a plan written down for the twisting of the points before it ever begins. That is the arc, then, from the underground to the institutional review board, from the desperate crossing to the funded protocol, from the molecule doing extraordinary things in the dark to the molecule, perhaps, doing them in the light, with someone watching. And so we return, one last time, to the spine of everything we have traced. The question was never really whether Ibogaine does something. It plainly does. The question was always how, by what braided, promiscuous, still unresolved mechanism, a single alkaloid from the root of a West African shrub reaches into the addicted brain and, in one long night, seems to loosen the grip of a dependence that no other molecule has ever loosened so quickly. And the honest answer, after everything, remains the one we began with. Nobody fully knows. Not the underground healers, not the Stanford psychiatrists, not the pharmacologists who have mapped every receptor it touches. We know the pieces: the mu-opioid partial interaction, the NMDA channel block that softens the glutamate storm of withdrawal, the serotonin transporter it inhibits, the sigma receptors it visits, the GDNF it seems to summon, that neurotrophic tide that may rewire the reward circuitry and hold the change in place long after the molecule is gone. We know the metabolism, the demethylation by the liver enzyme CYP2D6 into noribogaine, the long-lived child that lingers for days and keeps blocking hERG while the parent has departed. We know the peril, and now we know, at least in outline, one way to blunt it. But the synthesis, the single sentence that would explain how all these threads together produce an interruption, that sentence has not yet been written. The molecule interrupts addiction, and nobody knows how. That has been the refrain from the first minute, and it is still true in the last. What has changed is only this: that after half a century of shadow, someone has finally begun, carefully, with a monitor running and magnesium in the blood, to try to find out. Rest here a moment with that thought. Somewhere tonight, the fentanyl tide is still rising, still measured in micrograms, still pulling people under the surface of their own breathing. And somewhere else, in a hospital with a plan written down, a small and fragile science is trying to answer whether one long night with an old root alkaloid might be a hand reaching down into that water. Not a certainty, not yet a cure. A signal, bright, unproven, and watched at last. The promise and the peril no longer alone in the dark, but held now in the same careful room under the same steady light, with someone finally required to be certain that the trace is quiet, that the heart is holding its rhythm, that the tide of potassium still turns on time inside the chambers of a resting chest. A molecule at the edge of understanding, a crisis desperate enough to make us look. And the slow, patient, unfinished work of finding out, which is, in the end, the only thing that ever turns a mystery into medicine. So let us hold the whole of it, one last time, in the quiet. One dose, one day. And on the other side, for reasons we still cannot fully name, the opioid withdrawal quieted, the craving loosened, the compulsion that stole years simply stepping back from the door. 60 years from a Gabonese root to a Nature medicine paper, and still the honest answer to how is a shrug wrapped in a hypothesis. Think of the long arc of it now. The Bwiti initiates, walking into the dark with the root of Tabernanthe iboga on their tongues. Howard Lotsof, a young man in withdrawal, stumbling by accident onto a molecule that would take his hunger away. The tangled fistful of targets, the kappa and mu opioid receptors, the NMDA channel, the sigma sites, the serotonin and dopamine transporters, the alpha-3 beta-4 nicotinic receptor quietly closing. The noribogaine that lingers for days after the parent drug is gone, made by your own CYP2D6 and outstaying its maker. The unproven whisper of GDNF, a growth factor that might, or might not, reset the circuitry of wanting. And the shadow across all of it, the deaths, from a heart whose electrical rhythm was stretched too far, the QT interval widened until the beat lost its footing. The ban, the underground, the careful revival. Now come back to your own chest. Feel it, the slow, steady drum beneath everything. That rhythm is electrical, ions crossing membranes on schedule, potassium flowing out to reset each beat, the interval between them held to a faithful length. That same interval is the one this molecule can lengthen. The medicine and the poison live in the same place, in the same chemistry, told apart only by dose, by chance, and by the wide territory of what we still do not know. The mind quieting, the heart keeping time, both holding their fragile balance in the dark. Let your breathing slow. Let the questions rest unanswered. They will still be here, patient in the morning. There is nothing to solve tonight, only the steady beat, only the quiet, only the mind and the heart holding. Sleep well. Good night.